Hello! In this video, I'm going to talk about thalassemia, but like last time, I'm going to combine it with food. So this time I'm going to make dangos to try and give you an idea of the genetics and inheritance of thalassemia. So first, I'll be making the dangos first. And because I want it in 3 different colors, I have pumpkin, tofu, and matcha. So it'll be orange, white, and green. There's a lot of different recipes for dango, so I just kind of took a random one. While I'm peeling the pumpkin there, I'll give some background info about thalassemia. Thalassemia is a very common condition, especially in Southeast Asia, where Hong Kong belongs. And according to the Department of Health of Hong Kong, 1 in 12 of our population are carriers of either alpha or beta thalassemia. And that's probably because historically we used to have malaria, and thalassemia actually carries a protective effect against malaria. But of course, since the 60s, we don't have malaria anymore, which is a good news. Either way, thalassemia is quite common in our population. So what is thalassemia? Thalassemia is a disease due to decreased synthesis of either alpha or beta chain that make up the hemoglobin. Because hemoglobin is actually made up of 2 alpha and 2 beta chains, or at least most of the adult hemoglobins are. So we have decreased synthesis of these chains, but actually in thalassemia, the alpha and beta chain are structurally normal. We just have less of them. So we end up with less hemoglobin and less material, less of the raw material to make the red blood cell. Because we have less material, our red blood cells tend to be smaller and paler, and in more severe thalassemias, we also get anemia, which means that we simply don't have enough red blood cells. And the degree of the disease, how severe the anemia is, depends on how many of the alpha or beta genes are affected and also what kind of mutation they are. Here's my boiled doughs. All you need to do really is to boil the water, put these in the boiling water, and wait for it until it floats up. Now I'm going to assemble these mochi balls into dango, and I'm going to assemble it as if they were genes on a chromosome. First, I'll try to make the chromosome 16, which has genes that code for alpha globulin. And I'll represent the alpha globulin with the white balls. This is the zeta, which is a fetal globulin, but what we're interested in here is the alpha 1 and alpha 2. And by the way, this is the 5 end of the chromosome and this is the 3 end of the chromosome. I'll also thread one for the chromosome 11, which contains beta globulins. And just in case you forgot, a hemoglobin consists of 4 globulins, 2 alpha globulins, and 2 beta globulins. So unlike the alpha globulin, which has 2 genes coding for it, for beta globulin, we only have 1 gene. And I'll represent the beta globulin with an orange ball, like this. So all of these are fetal globulins involved for making fetal hemoglobin, and this one is a kind of a rare one. It's involved in making hemoglobin A2, which is present in very small quantities in adult. So normally what we have is the beta globulins with the alpha globulins. So 2 alpha and 2 beta globulin make up 1 the ingredients for one hemoglobin. Now let's look a bit more in detail at the genetics of alpha thalassemia. We already know that alpha thalassemia is due to mutation in the alpha gene. And we have two alpha genes in our chromosome 16, which both participate in coding for our alpha globulin. So in a normal human being, we have two of the normal alpha globulins. And because one chromosome is from our mother and the other is from our father, we end up with 4 alpha genes, like this. In alpha thalassemia carrier, what happens is one of our parents have all of the normal alpha genes, but from our other parent, we end up with a deletional mutation. Or sometimes we have non-deletional mutation, but it's the kind of mutation that don't give you any of the alpha globulins. So, effectively, we have 3 functional alpha genes. And so, when you have 3 normal genes, it's called alpha thalassemia carrier. With alpha thalassemia minor, it is when 2 genes are affected. So there's different configurations for the 2 genes. One is you can get both normal, one absolutely none. Another configuration is you can get 1 on each chromosome. Like so. But the thing is, this kind of thing is actually really rare in Chinese. So when we talk about alpha thalassemia minor, it's really about one chromosome being all normal, the other having none of the functional alpha genes. For HPH disease, we have one chromosome with the normal one and one with just none of the normal genes. So all like 3 deletions is HPH disease. Now, what is alpha thalassemia major or hydros fatalis in terms of genetics? Well, it's when all 4 genes are deleted, all 4 of the alpha genes. So we just have none of the functioning alpha gene, and that's why you get really severe anemia, and the baby usually can't survive past the fetal stage, so they just die in the womb. Since we talked about the genetics of alpha thalassemia, now I'll talk briefly about the inheritance. And it's really pretty simple. Because you know how we inherit one chromosome from our father and mother? It's basically how it works. So imagine we have a dad who has alpha thalassemia trait. So he's a carrier. The mom also has alpha thalassemia trait. And my question would be, what is the chance of the baby having alpha thalassemia major or hydros fatalis? We can use a Punnett square to help us. So imagine this is a column for the mom who has one chromosome with all of the alpha genes intact and one with none of the alpha genes. And dad, who is also a carrier, so one chromosome with all of the alpha genes intact, one with none. The chance of having a normal baby, like so, would be 1 in 4 according to the square. Now, what is the chance of the baby being alpha thalassemia carrier? From this Punnett square, we know that the baby can get the normal chromosomes from the dad or from the mom, and the defective from the mom or the dad. So he has 2 in 4 chance of having alpha thalassemia trait. Now, what is the baby having none of the alpha genes? We just need to complete this square. So he has 1 in 4 chance of having hydros fatalis if both of the parents have alpha thalassemia trait. Now I'll be talking about beta thalassemia. Beta thalassemia is quite different from alpha thalassemia because, as you just looked at, alpha thalassemia involves a deletional mutation, which means normally you have 2 alpha genes, but when you mutate, you end up with just 1 alpha gene on a chromosome, or even none, like this. But for beta thalassemia, first off, you only have 1 beta gene on chromosome, unlike the alpha, which you have 2. And also, when it mutates, it's not a deletional mutation. You just get a point mutation. So what happens in the point mutation is like you have a bit of a bleb on your gene, like this. You can see the green dot, right? So normal is supposed to be like all orange, but you get a point mutation, so you end up with a bit of a green. And what happens when it's like this is that you might get some beta globulin production still, but it might not be as much as it's supposed to. And the point mutations are actually quite varied. So depending on what kind of mutation you get, you have different severities. So for example, you can get a point mutation like having a green dot, or you can have a point mutation like having a white dot. And maybe this white dot ends up with a phenotype that's more severe than the green dot, so you get less globulin production. So in beta thalassemia minor, sorry for sticky hands, you have 1 normal chromosome with the normal beta gene, and 1 with a slight mutation. So, because remember, you have 1 chromosome from the mom and 1 chromosome from the dad. So you end up with slightly less than normal production of the beta globulins, but it's still okay. In beta thalassemia intermedia, it's also 1 normal it's also 1 normal beta gene, but the phenotype is more severe than the beta thalassemia minor just because the point mutation is one that is sort of worse, so you get even less beta globulin. But it's still really just 1 chromosome that has a mutated beta gene. What happens in beta thalassemia major is that most of the time it's compound heterozygous, which means that you have 1 kind of mutation on beta gene, but on the other chromosome you have another kind of mutation on the beta gene. But together, because both of them are a bit defective, you have quite severe anemia. And sometimes you also do get compound homozygous, but because there's a lot of different mutations on beta gene that can cause beta thalassemia, it's quite uncommon that you get compound homozygous mutation causing beta thalassemia major. I finished my Dango and I'll just put some honey on here. Yay! I pan-fried this because I've been playing with it for a long time and I probably contaminated it. And finally, time for food! Pretty good. Pretty chewy. I prefer this to gingerbread man. Thank you for watching the video. I hope you enjoyed it and learned something about thalassemia. Thank you.